All right, so at this point, we have talked about, we've made it all the way up through page 94 of your binder, and we've talked about how DNA gets transcribed into RNA and how RNA gets processed in eukaryotes through the process of a 5' cap, a poly A tail, and splicing. And then remember, in prokaryotes, there's no processing needed, it's already ready. And so what we're going to talk about next, this is on page 95. It says chapter 17, part 2 notes. What you're going to do is you're going to change that 17 to a 14 because this is actually chapter 14. 17 is from my old book. So in your current book, it's chapter 14, 2, translation. What happens to that mRNA when it's ready to be translated? All right, so here are the three components. mRNA is the message, tRNA is the interpreter, and the ribosome is the site of translation. So we had transcription occurring in the nucleus of the eukaryote and in the cytoplasm of the prokaryote. The prokaryote mRNA is ready to be translated. The eukaryote mRNA had to be processed. Then it leaves the nucleus and then it gets translated at the ribosome, and then tRNA is the interpreter. So if you look at this picture on the right, the ribosome is the brown thing, tRNA is the green, and then the growing polypeptide is the purple, and then the red is the mRNA that's getting translated. So I want to talk a little bit about tRNA, what that is. tRNA can be shown to you as a picture in a few different ways. My favorite way to look at tRNA is the two-dimensional shape because it's shaped like a T, and you can see this thing called an anticodon. I'll tell you what that is in just a minute. You can also see it in a three-dimensional structure. It looks like a jumbled mess right here, and then a really simple symbol used in this book. It looks, why they did a bent candlestick, I don't know. They could have at least done like a T. But anyhow, I didn't write the textbook, so you know, the bent candle with the wick hanging out, that is also tRNA. So all three of these are the same thing, just looked at either from a different angle or oversimplified just to uncomplicate things. All right, tRNA is transcribed in the nucleus. That just means it's made in the nucleus. It's made from DNA because it's RNA. So it has to be transcribed in the nucleus and then it leaves the nucleus. Each tRNA is specific to an amino acid. And so remember how many amino acids there are. There's 20 different amino acids. What they're going to do is transfer amino acids to ribosomes based on what the mRNA is coding for. All right. So if mRNA says something specific, tRNA is going to match up the anticodon to the codon, bring in the amino acid that is needed. All right. So the anticodon, there are these three letters down here. The anticodon is complementary to the mRNA codon. So if this anticodon is AAG, this tRNA actually matches up to UUC on mRNA. And the amino acid that it's going to be carrying is the amino acid from the amino acid chart that corresponds to UUC. So I need you to look at page 101. So pull out page 101. At this point in time in cell bio, you definitely should have used one of these codon charts. We will get some practice with it next time I see you, but I want you just to kind of refamiliarize yourself. What do you see when you look up UUC? So remember, you go left, top, right, wherever all three of those meet up, that's the amino acid. And I don't have my chart in front of me because I'm at home right now, but from what I can remember, UUC is supposed to code for phenylalanine, PHE. You don't need to have any of those memorized. There's actually only one that you need memorized. I'm going to tell you what it is in a minute, but you don't need to have that one. So this anticodon is going to match up with UUC. It's going to be carrying a phenylalanine, and it's going to drop phenylalanine off at the ribosome. So I said that there were 64 different codon options, but only 20 amino acids. will we have 64 different tRNAs? And the answer to that question is no. There's this thing called wobble. Wobble means the third base of the anticodon isn't very strict with what it matches with. And that's not a problem. I'm going to show you an example. So you see that this tRNA right here, it's the green thing, has the anticodon GUG. It's complementary to the codon GAG. The tRNA has the anticodon GAG. It is complementary to the mRNA codon CUC. G pairs with C, A pairs with U, G pairs with C. You'll see where I'm going with that. If you look on your amino acid chart on page 101, CUC is going to code for the amino acid leucine. All right, cool. What if you have the mRNA codon CUU? If you look on your amino acid chart, CUU also codes for leucine. I think there's actually about 5 different codons that all code for leucine. So why can't the GAG tRNA just go ahead and bring leucine in, even if that last G doesn't actually pair with that U? So I hope that's called wobble pairing. It's like, yeah, it's GAG, it doesn't completely match CUU, but we're just going to go ahead and use this tRNA for efficiency purposes because CUU codes for leucine anyways. So might as well just let that tRNA bring in that amino acid. So that's called wobble pairing. All right, there's an enzyme called aminoacyl tRNA synthetase. This is the enzyme that's going to help the tRNA bind to its amino acid that it's bringing in. So if you look at this gray thing, it looks kind of like an embryo. It really does. That looks like the eye and that looks like the little tail. Anyways, this embryo-shaped amino acid, there's no significance in it being shaped like that. It's just every time I see it, I'm like, okay, couldn't they have drawn it a little bit different? So this amino acid right here uses energy to take this enzyme right here takes the amino acid and uses energy to bind that amino acid to the tRNA that's carrying it. Sorry, I keep saying the wrong words. Let me say that again. This enzyme takes energy to bind the amino acid to the tRNA that it's matched to. And then now that tRNA is bound to its amino acid. So that's what that enzyme does. Just so you know, there's that enzyme and it does that. The ribosomes are made out of ribosomal RNA and proteins. So you have RNA with proteins. Ribosomes are made in the nucleolus. We've talked about that already. That's the very center of the nucleus. And there's 2 subunits. Remember I compared it to like a hamburger, like with a big bun and a little bun. So pause if you need to write that down because I'm going to move on. There are 3 sites. The ribosome has 3 sites. The A, the P, and the E. And I'm reading them from right to left because that's actually how this works. A, then P, then E. And you'll see what I mean in a minute. So that's the order that they go in. A, P, E. The A site is holding the amino acid that is to be added. So all you need to know is tRNA brings its amino acid into the A site. P site is holding the growing polypeptide chain. The E site is the exit. tRNA is going to leave once it drops off its amino acid. So basically, here's how it goes. tRNA is holding its amino acid, brings it into the A site. The amino acid that's on the tRNA in the A site then bonds to the growing amino acid. Then the tRNA and the growing polypeptide chain of amino acids, basically. Then the tRNA is going to move over to the P site. Then it's going to move over to the E site. Then it's going to exit, but it's going to leave that polypeptide chain behind. I know that's kind of confusing. So I will talk about that in just a little while. So as long as you have these 3 things written down, we're going to move on and then I'm going to go back to that because now so I've given you some background information, talked about what tRNA is, kind of how tRNA works, how it brings in an amino acid, it kind of matches up with the mRNA, what the wobble is, what the ribosome looks like. Now let's actually go through the really specific steps of translation. Okay, we've talked about all the parts. Let's go through the steps. Conveniently, the steps of translation are the same steps as they're named as transcription. Initiation, elongation, termination. Thank goodness. Only 3 steps: initiation, elongation, termination. But what happens in each of those steps is different, obviously, than transcription because it's achieving a different goal. What I want you to know about initiation is the start codon is AUG. The start codon is AUG. So when you see an AUG on mRNA, that is where translation starts. If you see an AUG, translation has started, translation is continuing, and you come upon another AUG, that doesn't mean translation starts over or that it stops. That just means, oh, we have a methionine that goes in that spot. So it's the start codon, but it also codes for an amino acid, methionine, that is the start amino acid, but it can also function as just a regular amino acid. Okay, so AUG codes for methionine, and you can check me. You can look at your chart on page 101. AUG, you should see methionine or MET right here. Okay, that's going to get everything started. It's going to, GTP is coming in. So all I need you to know is that all of this takes energy. I mean, it all takes energy, right? I mean, that's just kind of a given. But just in case you were wondering, GTP is the energy source used to get all of this assembled, right? So the small subunit binds to the start codon, tRNA carries methionine, it attaches to the P site, and then the large subunit attaches. So pause if you need to write that down because I'm going to click. Right, the second step of translation is called elongation. This is where the growing polypeptide becomes longer, basically. And so here are these steps that I was talking about. So if you look here in the P site, you have your growing amino acid chain. Then you see this other tRNA coming into the A site with its amino acid, right? So there's some codon recognition going on. The anticodon matches with the codon, brings in its amino acid, okay? So you're gonna probably want to label this on page 97. So there's a spot on page 97 where you can label all of these. So go ahead and pause. So if you see this little red arrow here where my cursor is, the long amino acid chain is now going to form a peptide bond with the new amino acid that just came in. And so you can see a peptide bond formation occurring between the new amino acid and the existing ones. And you see how the entire chain kind of shifted over to the right to that new tRNA that originally came in this first step. So you can just follow the steps down. This takes energy, obviously. So the new amino acid bonds with a peptide bond to the growing amino acid chain. And then both of these tRNAs shift over to the left. Okay? What was in the A site now shifts to the P. What was in the P site now shifts to the E. And the one that's now in the E site leaves. So then what happens after this? Well, you just go back to this original spot. A new amino acid comes in, peptide bond forms, tRNAs move to the left, the tRNA that drops off its amino acid leaves. All right? And I'll find a good animation for you, but let's just take the notes now, try to understand everything you can now, and then we'll do some reviewing and animations in class next time I see you. So 1st and 3rd, that'll be Monday, and then 6th, that'll be Tuesday. I won't give you any new material on Monday and Tuesday. Well, let's see, for 1st period, you won't have any new material on Monday. 3rd period, you won't have any new material on Monday. 4th period will, and then 6th period Tuesday won't. Whatever, you'll just see. All right, and then it's repeated over and over again until it reaches a termination sequence. The termination sequence in transcription, I told you didn't have to know. Translation, though, it's really easy to find the termination sequence. You just look at your amino acid chart. If you look at your amino acid chart, you see the word stop 3 times. And so if you look at what mRNA codons match stop, It's UAG, UAA, UGA. You don't have to have any of those memorized. The only one I want you to have memorized is AUG is start, and it goes for methionine. But the rest of these, you can just look at, and even like, you don't even need to know AUG's code for methionine because you look at that, but you need to know methionine would be the start, codon start amino acid. Right? So once that stop codon, one of those options is reached, a release factor binds and basically says, hey, ribosome, we're done. And so the ribosome disassembles, takes energy, and now you have your free polypeptides, so long and beautiful and ready to be folded. Okay? So that is termination, so pause if you need to keep writing. I just want to make a little, just a little note here. Under polyribosomes, poly means many, ribosomes are ribosomes. This is something that is done for efficiency. For efficiency. I want you to write the word efficiency next to polyribosomes. One mRNA can be translated at the same time by several ribosomes, all at the same time. So you have one mRNA, and in this picture, you have 1, 2, 3, 4 active ribosomes. So how many different polypeptides at this instant in time from this picture are you getting from these 4 ribosomes? One mRNA, 4 ribosomes, 4 polypeptides. That's so cool. So we don't need to transcribe DNA 4 times to get the 4 polypeptides. Just this one time because it's looking like a lot of proteins needed. So let's say you drink a big glass of milk and you haven't had milk in like a year. Your body stops making lactase. If you stop drinking or eating dairy products, your body's going to stop making lactase. Well, all of a sudden you're like, man, I just need some milk. Or let's say your doctor tells you, hey, you got to drink some milk or something like that. So you drink it after a really long time, introduce lactose, and your body's like, what? We got to digest this. So all of a sudden, the DNA that codes for the enzyme lactase gets transcribed, and then that mRNA is going to have many ribosomes on it, making lots and lots and lots of that protein. So this is for efficiency. So if you didn't get that, go ahead and get that down. Okay, let's talk about folding. We got our long, beautiful polypeptide. What are we going to do with it next? It needs to fold. So during synthesis, the polypeptide chain coils and folds spontaneously. And we've talked about this. There's our chaperonin again. I swear we've talked about this. Like, I really do believe that when I talked about proteins, I talked about chaperonins. The chaperonin is actually a protein itself. I think I compared it to a dressing room. So you go into J. Crew, you pick some stuff up off of the clothing rack and you want to try it on. You go into the dressing room. The dressing room itself does not help you. It just protects you from creepy people, right? This is kind of what the chaperonin does for the long polypeptide. It doesn't fold the polypeptide. It just protects it from outside influences and anything that can mess up the folding. And then the beautiful folded polypeptide leaves. Okay? Hopefully I didn't make that more confusing. So what came first? Well, if the chaperonin helps fold proteins, students always ask me this every year. What helped fold the first chaperonin? And I don't know that answer. I just don't. I'm sorry. It's like the chicken and egg question. All right, we have different types of ribosomes. This is going to be review. Free ribosomes synthesize proteins that stay within that cell. Bound ribosomes are going to make proteins that are to be secreted and exported from that cell. Counterintuitive. You would think free ribosomes, it's making free, they get to leave. No, free ribosomes are in the cytosol. They make proteins to be used for that cell. Bound ribosomes bound to the ER are going to make proteins that need to be packaged and processed and sent out of the cell for other cells to use. It could be signaling molecules. It could be like anything. They help each other out. All right? Make sure you get that down. Bound ribosomes, as they're packaging and processing proteins, are going to use something called a signal peptide. A signal peptide is added to the proteins that are going to be exported from the cell. So what you can think of a signal peptide as is like a zip code. So pause if you still need to write, because I'm going to click. A signal peptide is like a zip code. It's 20 amino acids long at the leading end of a polypeptide that's supposed to be exported from the cell. And it's like a zip code. And so if you look at this picture, so I would pause and write and then listen to the explanation. So if you look at the picture, starts here on the left. You have the ribosome, the mRNA, the signal peptide is in green. The long polypeptide's being made, okay? And the signal recognition particle brings the ribosome to the ER, and the protein is going to get excreted into the ER, and then it can get packaged and transported. So that signal peptide, that would have been kind of like a loose ribosome that's going to bring that polypeptide to the ER. So what I want you to kind of see here is like, wait a second, ribosomes bound to the ER, they're not always just sitting there. Ribosomes are going to always be in the cytosol. It's when there's a signal peptide that the ribosome moves over to the ER, binds to it, releases the protein within. So when you see rough ER, the rough part are ribosomes that are actively pushing polypeptides in. So what does this ribosome do when it's done? It leaves. And so it's almost like ribosomes are like transient, I guess. They move back and forth between the cytosol and the rough ER. So all I really want you to know is if there's this like the cellular zip code, the signal recognition particle that recognizes a code on the polypeptide that's like, whoa, this is about to be transported. Let me take you to the post office, basically. It's like the mailman. The mailman's going to get the letter from the mailbox and take it to the post office. If you have questions on that, make sure to note it at the end when I ask you the question about what you're confused on. All right, I'm going to talk briefly about mutations, and then we're going to talk more about mutations next week because it's really interesting topic that students seem to be really interested in. So we'll get all of this out of the way, lots of practice, and then talking about interesting mutations next week. So the central dogma is that DNA gets transcribed into RNA, RNA gets translated into a protein. Where do mutations actually happen? Here, during replication. So the first thing I want you to know about mutations is that they happen during replication, not transcription and translation. If a mistake is made in transcription or translation, no big deal. It just gets degraded by the cell, and then the cell's like, okay, I'll try again. But if there is a mistake made with DNA, that is when problems happen. Okay, so mutation is a change in the genetic material of the cell. We can have large-scale mutations or point mutations. For our purposes for this chapter, we are only going to talk about point mutations right now. So large-scale mutations that involve chromosomes, we're not going to touch on until we get to meiosis. So don't worry about those right now. So large-scale mutations would involve a whole chromosome, always causes disorder and death. These are the examples. We will touch on this again, though. So I would write it down just for completion's sake, but we are going to look at large-scale mutations when we talk about meiosis. Today, I want to focus on point mutations. All right, so pause, and if you need to finish writing this down, because I'm going to shift my attention to point mutations. A point mutation is just one base getting messed up. One base, one A, T, C, or G getting messed up. And so you can have what's called a substitution where one is replaced with another. Let's say there was an A and it got replaced with a T. Or let's say there was a C and it got replaced with a G, or any other combination. So there's two different types of substitution mutations. One's called a missense and one's called a nonsense. There's actually a third that's called a silent mutation. So a missense mutation is going to give you a different amino acid than what you're supposed to have because DNA is mutated, the wrong base is put in. Well, then mRNA is going to be wrong, then the protein's going to be wrong. Sometimes. So missense would be a different amino acid. Nonsense would be a premature stop codon is reached. And then a silent mutation is, yes, there was a DNA mutation. However, it just so happened to code for a codon that codes for the original amino acid that was meant to be there in the first place. Whew, so you get lucky. A frameshift mutation is going to be caused by an insertion or a deletion of a base, which is going to completely shift the reading frame of triplets, of codons, either to the left or to the right, will always cause non-functional proteins. So if I were to ask you, which is more serious, a substitution or a frame shift, you would need to say frame shift. That doesn't mean substitution can't lead to disease. It just means frame shift is going to not only change one amino acid, but every amino acid after that. All right. So substitution. So these are the examples that I'm referring to. This is under Veracross. This would be an example of missense. So you'll need to locate this under Veracross when you're studying. So I'm just going to show you some examples real quick. All right, here is your original on the top. There's your template, there's your RNA, there are your amino acids. All right, that's your original. It's called wild type. Wild type means like normal in this class. All right, now come down here. Your template strand now has a T in this spot instead of the C that was originally there. So now that means that RNA is going to have an A. That means that your RNA is going to be read AGC instead of GGC. So you get a serine instead of a glycine. That's a missense mutation. You get the wrong amino acid. Okay? A nonsense mutation. Let's look. This is the wild type up at the top. Here's your mutation right here. A is substituted for the T that was supposed to be there. So if you have an A in your template right there, you'll end up with a U. And then UAG, if you look at your amino acid chart on page 101, actually codes for stop. So that's called a nonsense because it's a premature stop codon. So instead of the lysine that was supposed to be there, it coded for a stop. That'll be a real messed up protein. So that's a nonsense. All right, here's a substitution. So here's your wild type at the top. Down here, you're supposed to have a G, but there's an A there instead. All right, so your mutation is there's an A instead of a G right here. So this triplet would be AAT. The mRNA would be UUA. I'm sorry, I'm looking at the wrong thing. So it's CCA up here. mRNA is GGU. It was supposed to be, mRNA was supposed to be GGC, coding for glycine. Now it's GGU, but because of the redundancy of the amino acid chart, it's still coding for glycine. So that's a silent mutation. A frame shift is going to, here's an insertion. So here's your wild type, here's your normal one. If you look where my arrow is, right here, this is where an A is getting inserted. So if an A gets inserted, the reading frame shifts to the left. So it's TAC, which codes for AUG, that's one codon. TTC right here codes for AAG, that's your second codon. Well, now everything is getting shifted to the left. So that AAG is no longer staying together. Now it's shifted, so it's UAA. So the reading frame gets shifted to the left. This one actually resulted in a premature stop codon. It could result in all different amino acids. We're going to do some practice on this next week, though. And then a deletion, same thing. If an A gets deleted, the reading frame then gets shifted over to the right because you're reading the codons 3 at a time, and then you end up with different amino acids. So that's called a frame shift mutation. All right, sickle cell is a result of a point mutation. It's a substitution. So here's your normal hemoglobin on the left. Here's your mutated hemoglobin on the right, just one base. There was supposed to be a T, now there's an A. So now RNA, instead of being GAA, it's now GUA, and now you have a valine instead of a glutamine, and that results in sickle cell disease. So that just shows you again what it looks like, and it results in a misshapen hemoglobin. It takes on a sickled shape. So the red blood cell takes on a sickled shape like down here. So this is normal up at the top, and then it takes on a sickled shape. So it's going to get stuck. It's not going to carry oxygen efficiently. Person with sickle cell anemia is going to be tired, and is going to have blood clots and stuff like that. All right, so last thing, I know this is going on kind of long. I just want you to have a summary of the differences between prokaryotes and eukaryotes, and then mutagens is there. Well, so under the word mutagen, a mutagen is just anything that can cause a mutation. So sickle cell disease is an example of a substitution mutation. A mutagen is anything that can cause a mutation. So like UV radiation can cause a mutation. Yeah. And then you're just doing a little difference between prokaryotes and eukaryotes. So I would go ahead and at least maybe on page 100 on the back, if you want to write down this list, these are the differences that we discussed. And so just kind of an overview. So it's really important to know the differences between prokaryotes and eukaryotes and the differences that they have in transcription and translation. All right, so instead of me reading this, I'm going to let you just write it down, and then you can go ahead and go. Like I said, next time I see you, I am just going to do a really thorough review of chapter 14. We're going to have a practice game, like a game you play on your own. We'll do a Kahoot. We'll do a practice worksheet just to make sure you really got this, so I'm not moving too fast through it. And then we'll do, we'll stay with chapter 14 for a few days, I think, because this is interesting stuff. And we'll do some stuff with the genetic mutations. All right, I will talk to you guys later.